So we're going to be doing a protein preparation. We've his tagged two proteins, an alpha body, two different alpha bodies with a nick and a his tag, and then we have a third protein, a pre, that does not have a nick. So we're going to be doing a protein preparation on these two, and then we're going to be seeing if we can co-purify our wild card protein over here. But for now, we're just going to start off with a generic spin down. This spin down can be used if you're doing a MIDI prep and you want to get a lot of DNA, or if you're doing competent cell preps, if you're doing a large scale chemical cell prep. But for now, we're going to be using this protocol for protein preparation. So you want to start off by labeling all of your tubes. These tubes are 250 mils, and we're going to be pouring our liquid in here. Now, a quick note about the origin of this culture. So what I did in order to do a protein prep is I put about 250 mils of media into this flask right here, and then I seed it in a 1 to 10. So I put about 25 mils of overnight culture in. I let that grow for an hour so that the cells can get in a mid-log phase and they can start dividing. And then after an hour, I induce with a rhabinose. Our expression plasmid is PBAD, so I induce with a rhabinose, and then I let it grow for an additional 4 hours. I actually let it grow for about 4.5 hours, so we should hopefully get a little bit more protein because of that. So the reason some people ask, well, why don't we just put a rhabinose right to begin with, or why don't you just put it on a constitutive promoter? So the problem with doing that is that sometimes the protein that you want to create is toxic, and you will not get any growth at all. So I've made the mistake of trying to induce immediately. I came back 5 hours later or overnight, and my culture was clear. So we let it grow up, and we let it get cloudy. We let the cells reproduce. So there's a ton of cells. So if the protein, which this one is a little bit toxic, it will have time to be expressed just a little bit. But since there's so many cells, we're going to get a lot overall. So now I'm just going to pour in my media, pour in my overnights, or my protein soup, and I'm going to pour it not all the way to the top, but just a little bit below the top, because when we centrifuge, I'm going to pour it right there, our liquid level goes from just being flat, it kind of angles a little bit, and if it's too high, it's going to leak out, and it's going to make a huge mess in the centrifuge, and that's just awful.
That's what it is.
So, yeah, and that's, if you ever have a smelly centrifuge, you can blame the person who's doing protein preps or MIDI preps. In our lab's case, that would be me. So I'm just going to pour these out. And they don't need to be perfect at this point. We're going to weigh them, and we have to balance the centrifuge so that it doesn't explode when we're running it. Because centrifuge explosions are just no fun. Okay, so kind of my first balance area is our technique for balancing is I put them all right next to each other, and I check the liquids, and they look like they're all about the same. If one is dramatically off, I will fix it right here. But we'll go into kind of the fine tuning. So I'm just going to cap them, and then we're going to move over to the centrifuge area. So I'm going to hold these tightly. And we're going to wait. So, bottle 1 weighs in at 279.1. Bottle 2 weighs in at 282.6. Bottle 3 weighs in at 279.6. So we have to bring them all up to the weight of this middle bottle. So we want them all to weigh 286 or 282.6. So I'm just going to add a little bit of water, or in my case PBS, just because that's sterile and we just need to add a little bit of weight. So in order to do that, I'm going to uncap our 279.1. I'm going to put it there, let it come up, and then I'm going to grab a pipette and some media. So, set my P1000 to a mil, pull off 5 mil. We want to go to 282.6, so 1, 2, and 3. And now we're going to start doing fine tuning. 282.3, 4, 5, 6. So this is now perfectly balanced. And it doesn't always need to be perfectly balanced, but my rule of thumb is you want to get it to within half a gram, or that's, I guess, the general rule of thumb. I like to get it in to 0.2 grams, and if it's dead on, that's the best. So, but you don't have to spend a half hour balancing, I guess is kind of the main point. So then we're going to balance this one as well. So we're at 279.6, so all we have to do is add 2 mils. 1. Apparently my math skills are off. We're going to add 3 mils. So 282.6. And now we are ready to spin. So I'll grab these bottles. I always want to double check that the tops are screwed on tightly. If we're to lose a top in there, it's going to change the weight, unbalance the centrifuge, and you can have a catastrophic collapse in centrifugal integrity. So right now, if you were to look in the centrifuge, you would see that we have a different type of rotor in place. This is a rotor for 96 well or 24 well format blocks that look like this. But our tubes will not fit in there. So I'm unscrewing. I'm going to pull this guy out. And we're going to replace it with this type of rotor, more normal looking rotor. I'm just going to plop it on top of that little axle right there. Screw it in. Make sure that it's really tight. And then, since I have 3 samples, I'm going to put my samples in a triangle. If I had 2 samples, they'd go across from another. If I had 4, they'd all go across from one another. And then I'm going to close this. And we have to change a couple things here. We want to change the rotor from S5700 to A10. So if we were to look at that serial number on that rotor, it's like TA10.125. We want A10. As for RPM, we're going to bump it up to 6500 for the pelleting set. And time is going to be 5 minutes. Oops. Press enter to set that information. Double check. RPM, rotor, time is 5. Enter again. Press start. And that's our spin down step. I usually like to hang out here for a little while and make sure that it's spinning down properly. But we'll be back after we've got a pellet.